Right now, at the bottom of the Mariana Trench, something is moving, not drifting, not floating. Moving deliberately, purposefully, through water that is cold enough to slow chemical reactions to a near stop, under pressure that would flatten a submarine, in a darkness so complete that the concept of vision has been abandoned by everything that lives there. Whatever is moving down there has never been seen by a human eye. It has never been photographed. It may never be named. And it is not alone. The Mariana Trench is not empty. It is not a dead zone. It is not the barren wasteland that every physical law of biology suggests it should be. It is alive in ways that science is only beginning to understand, in ways that break every rule that the study of life spent 2 centuries assembling. The rules said it should be impossible. The pressure alone 1,100 times the pressure of the atmosphere at sea level was supposed to be enough to end the question before it began. At that pressure, the proteins that run every biological process in every living cell undergo conformational changes that destroy their function. The enzymes that catalyze metabolism, the structural proteins that give cells their shape, the ion channels that allow nerves to fire, all of them are sensitive to pressure in ways that, beyond a certain depth, should make their operation impossible. And yet, at the bottom of the deepest place on Earth, life has not read the rules. It has not respected the physics. It has done something that the physics said it could not do, and it has done it for millions of years, in the dark, under conditions that would kill anything that evolved at the surface within seconds of arrival. What it has done, and how it has done it, is a story that starts not with the creatures themselves, but with the environment that shaped them. If you enjoy drifting into the deep ocean and discovering what lies beneath, please consider liking and subscribing. It truly helps more than you know. Are you ready? Let's begin. The Mariana Trench is not a single feature. It is a crescent-shaped depression in the western Pacific Ocean, stretching approximately 2,550 kilometers from end to end, formed where the Pacific Plate subducts beneath the Mariana Plate, where one enormous section of oceanic crust slides beneath another, forced downward by the collision of tectonic forces that operate on geological timescales. The trench is the direct result of this subduction. As the Pacific Plate bends and descends, it pulls the seafloor down with it, creating a scar in the ocean bottom that, at its deepest point, reaches approximately 10,935 meters below the surface. That number, 10,935 meters, is not intuitive. It requires context to understand. Mount Everest, the highest point on the surface of this planet, stands at 8,849 meters above sea level. If you placed Everest at the bottom of the Mariana Trench, its summit would still be more than 2 kilometers below the surface of the ocean. The trench is not merely deep. It is deep in a way that exceeds the scale of every other feature on this planet's surface. The pressure at that depth is a consequence of simple physics. Every meter of water above adds to the weight pressing down. At the surface, the atmosphere exerts approximately 101,325 pascals of pressure 1 atmosphere. Each 10 meters of seawater adds approximately 1 more atmosphere. At 10,935 meters, the total pressure is roughly 1,100 atmospheres, or approximately 1.1 times 10 to the power of 8 pascals. In practical terms, every square centimeter of surface area at the bottom of the Mariana Trench bears the equivalent of 1,100 kilograms of force pressing inward. A human being placed there without protection would not be crushed in the dramatic sense that the imagination conjures. The body is mostly water, and water does not compress significantly. What would happen instead is that the gas spaces, lungs, sinuses, digestive tract would be instantaneously obliterated, and the pressure differential between the internal gas spaces and the surrounding water would cause catastrophic tissue damage across every boundary between gas and liquid in the body. It would be over before the nervous system could process what was happening. The temperature at the bottom of the trench adds another layer to the hostility of the environment. The deep ocean is cold everywhere. The surface, solar heating that warms the upper ocean does not penetrate to the abyss, and the deep water is isolated from that warmth by density stratification that keeps cold, dense water at the bottom and warmer, lighter water above. At the bottom of the Mariana Trench, the temperature is approximately 1 to 4 degrees Celsius. Not freezing the pressure is too high for the water to freeze at those temperatures, but close enough to it that chemical reactions, which are temperature dependent, proceed at rates far below what surface biology considers normal. Metabolism at those temperatures, under those pressures, in complete darkness, with no photosynthesis to fix carbon from sunlight, it should not be able to sustain a community of organisms. And yet, the first time humans looked, they found one. The systematic exploration of the Mariana Trench is recent by almost any scientific standard. The first depth measurement suggesting the trench's existence was made in 1875 by the HMS Challenger, the vessel that gave the deepest point its name. The first direct observation of life at extreme depth in the trench came much later, from sampling equipment rather than cameras. The first crew descent to the Challenger Deep was made in 1960 by Jacques Piccard and Don Walsh in the bathyscaphe Trieste, a vehicle of extraordinary engineering that used gasoline for buoyancy and iron shot for ballast, and that descended to the bottom in roughly 5 hours. Piccard and Walsh were there for 20 minutes. They saw, through a small porthole, a flatfish-like organism moving across the sediment, and their observation was later disputed, with some scientists arguing the animal was more likely a sea cucumber. What is not disputed is that there was something alive down there. In the 6 decades since that first descent, the number of crewed visits to the Challenger Deep can be counted on one hand. James Cameron descended solo in 2,012 inches, the Deep Sea Challenger, spending approximately 3 hours at the bottom. Victor Vescovo made multiple dives in 2019 as part of the 5 Deeps expedition, reaching a new depth record of 10,924 meters. A handful of others have followed. The total time that human beings have spent at the bottom of the deepest place on Earth is measured in hours, not days. Hours. And yet, from those few hours of observation, and from the many more hours of uncrewed vehicle surveys and sediment sampling that have accompanied them, a picture has emerged. Not a complete picture. Nothing about the Mariana Trench is complete, and everything discovered there comes with a caveat that the surveys have covered a tiny fraction of the trench's total floor area. But enough of a picture to understand that the environment is not hostile to life in the way that the physics initially suggests. It is hostile to most life, and to the life that has evolved specifically for it, it is simply home. The first and most significant discovery was the composition of the sediment. The bottom of the Mariana Trench is covered in a layer of material that accumulates over geological time, a mix of marine snow, the constant rain of organic particles, dead organisms, and fecal matter that descends from the upper ocean, clay particles carried by deep currents, and the remains of microorganisms whose silica or calcium carbonate shells survive the long descent from the surface. This sediment is a food source. Not a rich food source by surface standards. The organic content is dilute, the particle flux is slow, and the energy available per unit area of seafloor is far less than what a coral reef or a kelp forest would provide. But it is there, and where there is organic carbon, there are organisms that have evolved to extract energy from it. The microorganisms at the bottom of the Mariana Trench represent some of the most pressure-adapted life forms on Earth. Studies of sediment from the trench have revealed bacterial communities at densities far higher than the surrounding deep ocean, because the trench is, in a sense, a collector. Its funnel-like geometry causes organic material that sinks into it to accumulate at the bottom rather than dispersing across the abyssal plain. The trench concentrates the food that falls from above. The bacteria that exploit this concentrated resource have evolved under extreme pressure for millions of years, and their biochemistry reflects that evolutionary history in ways that illuminate how pressure adaptation works at the molecular level. The key molecular adaptation is in the cell membrane. All cellular life uses membranes built primarily from phospholipid bilayers, two layers of fat molecules that separate the cell's interior from its exterior. These membranes must maintain a specific physical state, fluid enough to allow the proteins embedded in them to move and function, but structured enough to maintain the integrity of the cell. At high pressure, membranes tend to become more rigid. The pressure packs the lipid molecules more tightly, reducing fluidity. For a cell whose membrane proteins need to move, this rigidification is lethal. Deep-sea bacteria solve this problem by incorporating a higher proportion of unsaturated and polyunsaturated fatty acids into their membranes. These molecules have kinked structures that resist tight packing and maintain membrane fluidity even at extreme pressure. The same adaptation, it turns out, is present in every pressure-adapted organism in the deep ocean, from bacteria to fish. The membrane composition shifts systematically with depth, as if evolution has been running the same experiment independently in every lineage that colonized the deep and arriving at the same solution every time. What this reveals about the trench is something that the initial physical hostility of the environment obscures. The Mariana Trench is not inhospitable in the way that a vacuum or a fire is inhospitable. Inhospitable absolutely to everything, without exception. It is inhospitable to life that evolved for the surface, to life that has had time to adapt millions of years of selection pressure acting on organisms that survived while their neighbors perished. It offers something that no surface environment offers: stability. The temperature at the bottom of the Mariana Trench has not changed significantly in millions of years. The pressure has not changed. The chemistry of the water, within the limits imposed by the slow delivery of organic material from above, has been consistent. For organisms adapted to those conditions, the trench is not a crucible. It is a refuge. Stable, dark, cold, and deeply pressurized, but a refuge nonetheless. That stability has consequences for the evolutionary biology of deep trench organisms that are only beginning to be understood. In surface environments, climate change, sea level fluctuation, temperature swings, and the periodic disruption of food webs by mass extinction events drive rapid evolutionary change. Species go extinct, new ones emerge, communities restructure. At the bottom of the Mariana Trench, those disruptions are muted. The isolation of the deep trench environment means that lineages that colonized it early have had time to diverge from their surface relatives in ways that accumulate over geological time without the interruptions that reset evolutionary clocks at the surface. Some of the organisms in the deep trench may be among the most phylogenetically isolated life forms on Earth, separated from their nearest relatives, not by geography, but by depth, by pressure, by a gradient of physical conditions that most organisms cannot cross in either direction. But the most dramatic evidence of life in the Mariana Trench does not come from bacteria. It comes from animals. And the animals that have been found in the trench, the ones whose existence the physics of the deep ocean initially seems to preclude, are where the story of extreme depth life becomes genuinely strange. Because what lives at the bottom of the Mariana Trench is not a collection of barely surviving biological curiosities clinging to existence at the edge of the possible. What lives there is, in some cases, thriving. And understanding how that thriving is possible requires going deeper into the biology, into the specific molecular and physiological mechanisms that allow multicellular animals to function at pressures that, by every measure available to surface biology, should destroy them. The Mariana Trench did not always exist in its current form. The subduction zone that created it has been active for tens of millions of years. But the specific geometry of the trench, its depth, its width, its internal topography has changed continuously as the Pacific plate has moved and the subduction angle has varied. The deepest points of the trench are not uniform. There are multiple deep basins within the larger structure, separated by internal ridges, each with slightly different water chemistry, sediment composition, and biological communities. The Challenger Deep is the deepest of these basins, but it is not the only one, and the organisms in different basins within the same trench are not necessarily the same. The trench is not a single habitat. It is a collection of related but distinct habitats, each shaped by the specific physical and chemical conditions of that basin, each host to its own community of organisms that have evolved in relative isolation from the communities in adjacent basins. This internal heterogeneity of the trench is one of the reasons why sampling efforts have consistently returned new species even in areas that had been sampled before. The trench floor is not uniform terrain. It has ridges and valleys, soft sediment plains and hard rock outcrops, areas where water chemistry is influenced by the slow seepage of fluids through the sediment, and areas where it is not. Each microhabitat within the trench represents a slightly different selective environment, and the organisms in each have had millions of years to diverge from the organisms in adjacent microhabitats. The result is a diversity that the trench's extreme conditions would not initially suggest, not the low diversity of a harsh, simple environment, but something more complex and more biologically interesting than the physics of the place seems to allow. The sediment at the bottom of the Challenger Deep has been sampled and analyzed in detail by several research expeditions, most recently in connection with the Five Deeps expedition. What those analyses have found in the sediment is a microbial community of considerable complexity, dozens of distinct bacterial and archaeal lineages, many of them novel, many of them closely related to organisms found nowhere else on Earth. The density of microbial cells in the trench sediment is, as noted, higher than in the surrounding abyssal plain. The trench concentrates organic material, and the microbial community has expanded to exploit it. But the chemical signatures of that microbial activity are also unusual. Isotopic analyses suggest that the microorganisms in the trench sediment are cycling carbon and nitrogen through pathways that have been modified by the pressure environment, running metabolic processes at rates and through mechanisms that differ in measurable ways from the same processes in surface organisms or even other deep-sea microorganisms. What this means, biologically, is that the Mariana Trench is not simply an extreme version of the rest of the deep ocean. It is a distinct environment that has driven the evolution of distinct biology, not just extreme pressure tolerance added onto otherwise standard deep-sea biology, but a genuinely different biochemical approach to being alive. The organisms that live there are not merely surviving. They have, over millions of years of selection, been rebuilt from the biochemical level up to function in conditions that their evolutionary ancestors would not have survived for an hour. The question of how that rebuilding happened, what specific changes in what specific molecules produced organisms capable of thriving at 1,100 atmospheres, is one that biology is still actively investigating, and one that the limited access to the trench makes extraordinarily difficult to answer. Every answer that has emerged so far has come with another question attached. The membrane adaptation that maintains fluidity under pressure is understood in outline, but the specific lipid compositions of hadal organisms vary in ways that suggest the solution is not a single universal strategy, but a family of related strategies, each tuned to the specific pressure range and temperature profile of the organism's particular depth. The protein stabilization mechanisms that prevent pressure-induced unfolding are understood in broad terms, but the specific molecular changes that produce pressure-stable versions of common enzymes are still being mapped, one protein at a time, in a small number of species from a small number of samples. The full biochemical inventory of life at the bottom of the Mariana Trench has not been assembled. It probably cannot be assembled with current technology and current levels of access to the trench. What exists now is a series of snapshots, detailed, scientifically rich, but covering only a tiny fraction of the total biological diversity that the trench contains. And beneath all of this, beneath the microbial diversity, the lipid adaptations, the pressure-stable enzymes, the concentrated organic sediment is the more fundamental question that the Mariana Trench keeps raising every time anyone looks at it carefully. The question is not whether life can survive under extreme pressure. The trench has answered that question definitively. It can. The question is what that survival means for our understanding of where life can exist and where it might be found in environments that we have not yet examined. Because if life can thrive at 1,100 atmospheres, in total darkness, at temperatures near freezing, with no sunlight and no connection to the photosynthetic food chains that sustain almost everything else on this planet, then the range of environments where life is possible is significantly larger than the range of environments where we have been looking for it. And the specific mechanisms that make the Mariana Trench habitable, the molecular adaptations, the biochemical strategies, the evolutionary solutions that deep trench life has assembled over millions of years are not curiosities. They are a window into something fundamental about how life works and how far it can be pushed before it stops working. Understanding those mechanisms is the task that the next part of this story begins. The animals of the Mariana Trench did not arrive there recently. The Hadal Zone, the name given to ocean depths below 6,000 meters, found almost exclusively in the great oceanic trenches, has existed for tens of millions of years, and the lineages of animals that now inhabit it have had that entire span of geological time to adapt to its conditions. What they have produced in that time is a suite of biological solutions to the problem of extreme pressure that are distinct from anything found in shallower water, not incremental modifications of deep-sea biology, but genuine evolutionary innovations that appear specifically in hadal organisms and nowhere else. The most abundant animals at the bottom of the Mariana Trench are not the organisms that make the most dramatic impression. They are not the large, visually striking creatures that cameras tend to find and film. They are amphipods, small crustaceans, superficially similar to shrimp, ranging from a few millimeters to several centimeters in length, that exist in the Hadal Zone in densities that no one predicted before the first baited traps were lowered to the trench floor. When researchers deploy a trap baited with fish or meat at hadal depths and retrieve it several hours later, they typically find thousands of amphipods inside. Not dozens, thousands. The Mariana Trench, which the physical conditions suggest should be sparsely populated at best, turns out to be home to vast swarms of these small crustaceans, crowding around any available food source with an efficiency that reflects millions of years of evolution in a food-limited environment. The hadal amphipods, several species, belonging primarily to the genus Hirondelia and related genera, represent one of the clearest examples of hadal specialization in the animal kingdom. Their bodies are adapted to the pressure in ways that are now well documented. Their cell membranes, like those of the hadal bacteria, contain elevated proportions of unsaturated fatty acids. But the specific lipid compositions of hadal amphipods differ from those of their shallow water relatives in ways that go beyond simple membrane fluidity adjustment. Analysis of amphipods collected from different depths within and around the Mariana Trench has found that the lipid composition of their tissues changes systematically with depth, suggesting that the adaptation is not a fixed trait, but a continuous response to the specific pressure at the organism's particular depth. The same amphipod genome, expressing different lipid synthesis genes at different depths, producing different membrane compositions, a flexible molecular response to a varying physical environment. The protein chemistry of hatoamphipods is equally revealing. Proteins are pressure sensitive. The same conformational changes that make them functional can be reversed by pressure, unfolding the protein and destroying its activity. Deep-sea organisms deal with this problem through two mechanisms. The first is the evolution of intrinsically pressure-stable protein variants, versions of common enzymes, whose amino acid sequences have been modified by natural selection to resist the conformational changes that pressure induces. The second is the accumulation of small organic molecules called piezolites, the most important of which, in animals, is trimethylamine N-oxide, or TMAO. TMAO is a molecule that has been known to biochemists for decades as a metabolic waste product in surface fish, excreted through the gills and largely irrelevant to function. In deep-sea fish and invertebrates, TMAO has a completely different role. It acts as a molecular chaperone for proteins, a small molecule that preferentially associates with the water surrounding proteins and alters the thermodynamic properties of that water in ways that stabilize the proteins folded configuration against the destabilizing effects of pressure. The higher the pressure the organism lives under, the more TMAO its tissues contain. The relationship is so consistent that TMAO concentration in fish tissue has been proposed as a depth gauge, a way to estimate the depth at which a fish lives from the chemistry of its flesh, without knowing anything else about where it was caught. In hatoamphipods, TMAO concentrations are extraordinarily high, higher than in any other group of marine animals. The tissues of amphipods from the Mariana Trench contain TMAO at concentrations that would be toxic to surface organisms, but that are precisely calibrated to the pressure at which the amphipods live. The TMAO does not prevent pressure from acting on the proteins. It cannot do that. Pressure is a thermodynamic reality that permeates every molecule in the organism's body. What it does is shift the equilibrium between the folded and unfolded states of the proteins toward the folded state, counteracting the pressure-induced tendency toward unfolding. The protein remains functional. The enzyme continues to catalyze its reaction. The cell continues to do what cells do. The TMAO discovery has implications that extend well beyond the Mariana Trench. It suggests that the deep ocean has independently solved the problem of pressure-induced protein destabilization with a molecule that surface biology had dismissed as a minor metabolic product, and that the solution is elegant, enough to have evolved repeatedly in multiple hatolineages, converging on the same chemical strategy from different evolutionary starting points. It also suggests that the depth limit of animal life in the ocean may be set, at least in part, by the TMAO concentration limit, by how much of this stabilizing molecule an animal can accumulate in its tissues before the TMAO itself begins to cause other problems. Calculations based on measured TMAO concentrations in haddlefish and the relationship between TMAO and depth suggest that the theoretical maximum depth for fish life set by TMAO chemistry is approximately 8,200 meters. Above that depth, TMAO can stabilize proteins adequately. Below it, the required TMAO concentration would exceed the physiological tolerance of the organism. This prediction has since been tested by camera surveys at multiple depths within and around the Haddle Zone. The deepest confirmed fish observation, a snailfish filmed at 8,336 meters in the Izu Ogasawara Trench, sits close to but does not exceed the theoretical TMAO limit. The fish was behaving normally, swimming, foraging at home. The depth was consistent with the biochemical prediction right at the margin where TMAO chemistry can still hold proteins together against the pressure that would otherwise disassemble them. Below that margin in the deepest parts of the Mariana Trench, below 8,200 meters fish have not been found. Not because cameras haven't looked, because the TMAO limit predicts they cannot be there, and the cameras have confirmed the prediction. What lives below 8,200 meters in the trench is not vertebrate life. It is invertebrate life amphipods, sea cucumbers, polychaete worms, foraminifera organisms whose biochemistry manages the pressure problem through different mechanisms, or whose simpler body plans make fewer demands on pressure-sensitive protein systems. The transition from a fauna that includes fish to a fauna that does not is not abrupt. It happens gradually as depth increases through the range where the TMAO limit applies, but it is real, and it is visible in the data from baited camera surveys that have been conducted at multiple depths within the trench. The sea cucumbers that live at the bottom of the Mariana Trench deserve particular attention because they represent an extreme of Haddle adaptation that is rarely discussed outside of specialist literature. Sea cucumbers holothurians are echinoderms, relatives of starfish and sea urchins, soft-bodied animals that move slowly across the seafloor, ingesting sediment and extracting organic matter from it. At shallow depths, they are unremarkable, common, diverse, ecologically important, but not particularly dramatic. At the bottom of the Mariana Trench, they are dominant. Surveys of the trench floor have found that holothurians make up a substantial fraction of the visible megafauna, the animals large enough to be seen by camera, and that they appear to be actively feeding, moving, and functioning in an environment that should, by surface standards, be completely hostile to soft-bodied, slow-moving animals with no rigid skeleton to resist the pressure. How they do it is a question that has not been fully answered. The sea cucumber body plan has no gas-filled spaces, no swim bladder, no lungs, no air pockets of any kind. The body is effectively a bag of fluid with a few internal organs, separated from the surrounding water by a layer of muscle and connective tissue. Without gas spaces to be compressed, sea cucumbers face a simplified version of the pressure problem. They need their biochemistry to function under extreme pressure, but they do not face the additional challenge of managing collapsing air cavities. The TMAO mechanism and the membrane lipid adaptations that work for amphipods presumably work for holothurians as well. And the soft body plan that seems like a disadvantage in a high-pressure environment turns out to be an advantage. There is nothing to compress, nothing to implode, nothing that fails suddenly at a specific pressure threshold the way a gas bladder fails in a fish. The polychaete worms found at hadal depths represent yet another adaptation strategy. Polychaete segmented worms are among the most diverse and most ancient animal groups on Earth, present in virtually every marine environment from shallow coastal sediments to the deep abyss. The hadal polychaetes are not a single lineage, but multiple independent colonizations of the hadal zone by worm lineages from shallower depths, each of which has independently evolved the necessary pressure tolerances. The convergent evolution of pressure adaptation in polychaetes mirrors what is seen in amphipods and bacteria, the same molecular solutions appearing independently in multiple lineages, as if the problem of extreme pressure has a limited number of viable solutions and evolution reliably finds them given sufficient time. What all of these animals have in common, amphipods, sea cucumbers, snailfish near their depth limit, polychaete worms, is that none of them are recent arrivals at depth. All of them belong to lineages that colonized the hadal zone over evolutionary timescales, and all of them carry the molecular signatures of that long colonization. The elevated TMAO, the modified membrane lipids, the pressure-stable enzyme variants. They are not organisms that happened to survive when they sank too deep. They are organisms that evolved specifically for the environment they inhabit, over millions of years of selection in one of the most extreme physical environments on this planet. And the diversity of what has been found in the handful of trench surveys conducted to date, covering only a fraction of the trench's total floor area, is significant precisely because the surveys have been so limited. Every new survey of a previously unsampled section of the trench floor returns new species. Every new depth record for a known group of organisms revises the understanding of how deep that group can go. The TMAO depth limit for fish was not a prediction that existed before fish were found near the limit. It was a calculation made after the fish were found that then correctly predicted where fish would not be found. The biology of the hadal zone is not a closed book. It is a book that has barely been opened, and every page that has been read has contained something that required revision of the pages that came before. What has not yet been adequately explored, what the biology of the trench has been pointing toward without yet arriving at, is the question of what the trench floor looks like, not from the perspective of individual organisms, but from the perspective of an ecosystem. How does energy flow through a community of organisms in a place where the only energy input is the slow rain of organic material from above? What are the predator-prey relationships, the competitive hierarchies, the mutualistic dependencies that structure a community of hadal organisms? These are questions that require sustained observation over time, the kind of observation that has been possible in shallow marine environments for decades, but that the difficulty of access to the hadal zone has made nearly impossible to conduct there. The foraminifera single-celled organisms that build shells of calcium carbonate or agglutinated sediment particles deserve mention here, because their presence at hadal depths illuminates something important about the evolutionary history of the trench's inhabitants. Foraminifera are among the oldest eukaryotes, with a fossil record extending back over 500 million years. The species found in the Mariana Trench sediment are not identical to those found in shallower deep-sea sediments. They are hadal-specific variants, with shell structures and chemical compositions that differ from their shallow water relatives in ways consistent with pressure adaptation. The foraminifera in the trench sediment are also unusually large by the standards of their group, some reaching centimeter scale, at the upper limit of single-celled life, and their size may itself be an adaptation to the food-limited environment of the trench, where a larger cell volume provides more internal storage for the organic material that is metabolized slowly under low-temperature, high-pressure conditions. The xenophyophores, giant single-celled organisms that build elaborate structures from sediment particles and can reach tens of centimeters in diameter, are among the largest single-celled organisms on Earth, and they are found at the bottom of the Mariana Trench in numbers that were not expected. A xenophyophore is a single cell, not a colony of cells, a single, enormous cell with thousands of nuclei, managing its internal processes across a diameter that can exceed 20 centimeters. Their presence at hadal depths suggests that the constraints on cell size that apply in surface environments, constraints related to the efficiency of diffusion across large cell volumes, are modified by the specific chemical and physical conditions of the trench in ways that remain poorly understood. The bacteria that dominate the microbial community of the trench sediment are not passive participants in the ecosystem. They are the primary processors of the organic material that arrives from above, breaking down complex organic molecules into simpler compounds that other organisms can use, cycling nitrogen and sulfur through transformations that make these elements available for biological use, and producing metabolic byproducts that alter the chemistry of the sediment and the overlying water in ways that shape the environment for every other organism in the community. The hadal microbial community is, in this sense, the foundation of the hadal ecosystem, not just one component among many, but the metabolic engine that makes all the other components possible. This foundation depends entirely on the rain of organic material from the surface ocean. If the surface productivity that drives the biological pump, the process by which photosynthetically fixed carbon sinks to the deep ocean as dead organisms and fecal matter, were to decline significantly, the hadal ecosystem would be affected within the timescales of deep ocean circulation. The trench is isolated from the surface, but it is not independent of it. It is the downstream terminus of a flow of energy that begins with sunlight hitting the surface of the ocean thousands of meters above and passes through dozens of biological transformations before arriving at the sediment of the Challenger Deep. Understanding the hadal ecosystem in full requires understanding that entire chain from the phytoplankton at the surface to the bacteria in the sediment at the bottom and every trophic level in between. That understanding does not yet exist. The chain has been traced in outline, but the specific rates and efficiencies at each step are known only approximately. What is known is enough to establish that the hadal ecosystem is real, structured, and more biologically complex than the physical conditions suggest. What is not yet known is the full extent of that complexity and what it might reveal about the capacity of life to organize itself in the most extreme environments on this planet. The answers to those questions are waiting at the bottom of the Mariana Trench. And the specific mechanisms that organisms there have evolved to survive not just the membrane lipids and the TMAO and the pressure-stable enzymes, but the ecological relationships, the flow of energy through the community, the way life organizes itself when everything surface biology takes for granted has been stripped away to point towards something that the next part of this story explores. Because the Mariana Trench is not the only place where those conditions exist, and the organisms that have solved the problem of living there may be offering a preview of solutions that life has found or will find in environments that are much farther from this planet than 11 kilometers down. The Mariana Trench sits in the western Pacific, surrounded on all sides by open ocean. The nearest land of significance is Guam, approximately 200 kilometers to the east. The nearest shallow continental shelf is thousands of kilometers away. The trench is, in the most literal geographic sense, isolated a deep scar in the ocean floor, surrounded by abyssal plain, cut off from every other hadal environment on the planet by thousands of kilometers of open water, and the continuous gradient of depth and pressure that separates the trench floor from any other extreme pressure environment. The organisms that live in the Mariana Trench did not walk there. They did not swim there from adjacent trenches. They arrived in evolutionary time by descent through the water column from shallower depths, generation by generation, as natural selection pushed lineages deeper and deeper into the water, favoring the variants that could tolerate slightly more pressure than their ancestors, until the lineage had traversed the full gradient from moderate depth to hadal depth over millions of years. This process, the colonization of the hadal zone by descent through the water column, has not happened once. It has happened many times, independently, in many different lineages. The amphipods in the Mariana Trench are related to, but genetically distinct from, the amphipods in the Kermadec Trench south of Tonga, the Tonga Trench itself, the Puerto Rico Trench in the Atlantic, and the other major hadal systems scattered across the ocean basins. They share common ancestry, but they have been separated by the depth and distance between trenches long enough to have diverged into distinct species, sometimes distinct genera, with their own specific adaptations to the specific conditions of their particular trench. The hadal zone is not a single habitat inhabited by a single community. It is a collection of isolated habitat islands, each with its own endemic fauna, connected to each other only by the shallow water relatives of the organisms that colonized each trench independently. This biogeography has implications that are only beginning to be explored. Each trench represents a separate evolutionary experiment, the same basic problem of extreme pressure being solved by the same basic suite of molecular mechanisms, but with the specific details varying from trench to trench as the populations diverge in genetic isolation. Comparing the adaptations of hadal organisms across different trenches offers a way to identify which aspects of the pressure adaptation solution are universal, appearing independently in every trench because there is no alternative, and which are contingent, varying from trench to trench, because the specific evolutionary history of each trench's colonizing population happened to find slightly different solutions to the same problem. This comparative approach to hadal biology is one of the most productive avenues of current research, and it depends on having good biological surveys of multiple trenches, a condition that is only beginning to be met as the technology for hadal exploration improves. The Tonga Trench, located in the southwestern Pacific, is the second deepest trench in the world, reaching approximately 10,880 meters at its deepest point. Its amphipod community has been studied in some detail, and the comparison with the Mariana Trench fauna is revealing. The two communities share higher-level taxonomic groups, but are composed of different species, with different size distributions, different depth ranges for similar taxa, and different relative abundances of the various groups that constitute the community. The Tonga Trench fauna is not simply a copy of the Mariana Trench fauna transposed to a different location. It is a distinct community with its own evolutionary history, shaped by the specific colonization history of that trench and the specific physical conditions, depth, temperature, water chemistry, organic input rate that characterize the Tonga system. The Kermadec Trench, adjacent to the Tonga Trench in the same general region of the Pacific, presents a further comparison. It reaches approximately 10,047 meters and has been surveyed by multiple expeditions, including some of the most technically sophisticated hadal surveys conducted to date. The Kermadec fauna overlaps with, but is distinct from, both the Tonga and Mariana communities. Some species appear in multiple trenches, presumably because they are more recent colonists who have not yet had time to diverge into separate species, or because they retain some capacity for dispersal between trenches at intermediate depths. Others are confined to a single trench system, having been there long enough that the barriers to dispersal have allowed full speciation. The pattern of shared and endemic species across the hadal trenches is, in miniature, a snapshot of the evolutionary process itself of populations diverging under geographic isolation, accumulating genetic differences at a rate set by mutation and genetic drift, until the differences are great enough that the populations would no longer interbreed even if they came into contact. The depth gradient within individual trenches adds another dimension to this picture. Within the Mariana Trench, the fauna at 7,000 meters is not the same as the fauna at 9,000 meters, which is not the same as the fauna at 10,900 meters. The pressure difference between these depths, approximately 200 to 400 additional atmospheres, is large enough to impose significant physiological constraints, and the organisms at each depth level have adapted to the specific pressure at that level. The result is a vertical zonation within the trench. Different communities at different depths, each adapted to the pressure range of its particular depth zone, with transition zones between them where species from adjacent depth ranges overlap and compete, This vertical structure, which mirrors in miniature the broader vertical zonation of the entire ocean, from the sunlit surface through the twilight zone to the abyss to the hadal, is one of the features of trench biology that makes the hadal zone more complex than a simple extreme pressure variant of the deep ocean. The currents that flow through and around the Mariana Trench complicate the biogeography further. The deep Pacific is not a static body of water. It is traversed by sluggish but real currents, the thermohaline circulation that moves cold, dense water from the polar regions toward the tropics along the ocean floor, and the more local currents generated by the topography of the ocean bottom as deep water flows around and between features. These currents carry organic particles, the marine snow that feeds the hadal community from the surface production zones above toward the trench floor. They also carry larval and juvenile organisms. Many deep-sea invertebrates, including some hadal species, have planktonic larval stages that are dispersed by currents before settling at depth. The extent to which hadal populations are connected to each other through larval dispersal versus being fully isolated and evolving independently is a question that has not been definitively resolved for most hadal species. Genetic analyses of hadal amphipod populations from different trenches suggest that some gene flow occurs between trenches, implying that dispersal at intermediate depths occasionally connects populations that are otherwise separated by the full depth of open water. Other analyses suggest complete isolation between trench populations, even within the same ocean basin. The resolution of this question, whether hadal communities are evolutionary islands or something more connected, will require genetic sampling of a breadth that the difficulty of access to multiple trenches has so far made impossible. But the technology is improving, the sampling is expanding, and each new dataset adds another layer of resolution to a picture that has been blurry since the first baited trap came back full of amphipods from the bottom of the deepest place on Earth. The physical environment of the Mariana Trench also includes features that are not present in most deep-sea environments and that significantly affect the biology of the organisms living there. The subduction zone that created the trench is still active. The Pacific Plate is still sliding beneath the Mariana Plate, still bending and descending, still generating the geological forces that maintain the trench's depth against the tendency of sedimentation to fill it in. This ongoing geological activity means that the trench floor experiences periodic disturbances, small earthquakes caused by the movement of the plates, changes in sediment distribution caused by the slope instabilities that earthquakes trigger, and the slow but continuous deformation of the seafloor itself as the subducting plate reshapes the geometry of the trench from below. These geological disturbances affect the biology. Sediment slumps triggered by earthquakes can redistribute the organic material on the trench floor, concentrating it in new locations or burying it too deeply for surface-dwelling organisms to access. The earthquakes themselves produce pressure waves, seismic waves that travel through the water column and the sea floor, and whose effects on the organisms living there are largely unknown. Whether hadal organisms have specific adaptations to the episodic seismic disturbances of the subduction zone environment is a question that has barely been asked, let alone answered. The intersection of geology and biology in the Hadal Zone is a frontier that is almost entirely unexplored. There is one more physical feature of the Mariana Trench environment that deserves attention, because it affects the distribution of life within the trench in ways that have been observed but not fully explained. The bottom water of the Mariana Trench is not chemically uniform. Measurements at different locations within the Challenger Deep have found variations in dissolved oxygen content, pH, and the concentrations of various dissolved ions that suggest the bottom water is not well mixed, that different parts of the trench floor are bathed by water with slightly different chemical compositions. These chemical gradients are subtle compared to the variations that drive, say, the diversity of hydrothermal vent communities. But in an environment where organisms are adapted to very specific chemical conditions, even subtle variations can be biologically significant. The distribution of different species within the trench, why some are found only in certain basins, why some are more abundant in the eastern part of the trench than the western, may be partly a function of these chemical gradients, rather than, or in addition to, the pressure differences between depths. What the biogeography and physical environment of the Mariana Trench add up to, when considered together, is a picture of a place whose biological complexity is not accidental. Every feature of the environment, the isolation, the depth gradient, the geological activity, the chemical heterogeneity, the pattern of organic input has shaped the biology in specific, traceable ways. The trench is not simply a deep hole with some organisms in it. It is a structured habitat with a structured biological community, a community whose structure reflects millions of years of interaction between the physical environment and the organisms that have evolved within it. Understanding that structure, mapping it in the detail that ecology requires, is a project that has barely begun and that faces obstacles of access and technology that are genuinely difficult to overcome. But the obstacles are not impossible. They are the same kind of obstacles that faced the exploration of every other extreme environment that science has eventually characterized. The deep sea in general, the polar regions, the high atmosphere. Each of those environments was, at one point, as inaccessible and as mysterious as the Hadal Zone is today. Each was eventually characterized by a combination of technological development, sustained investment, and the accumulated effort of many researchers over many decades. The Hadal Zone is not fundamentally different. What it requires is time and tools and the sustained attention of a scientific community that has historically found other environments more accessible and more tractable. The attention is growing. The tools are improving. And what they are finding, as the access gradually increases, is a place whose biological richness continues to exceed what anyone predicted, not despite the extremity of the conditions, but in some sense because of it. The question of why the trench is so biologically rich, given how extreme the conditions are, has an answer that the biogeography and ecology only partially supply. The full answer requires going deeper not into the trench itself, but into the evolutionary history of the organisms that colonized it. Because the lineages that succeeded in the Hadal Zone did not succeed by accident. They succeeded because they possessed, before they ever descended to Hadal depths, certain biological characteristics that made the descent possible, characteristics that had evolved for other reasons in other environments, and that turned out to be pre-adaptations to the conditions of the deep trench. Identifying those pre-adaptations and understanding how they became the foundation for the full suite of Hadal adaptations that developed later is one of the central questions of Hadal evolutionary biology. The amphipods are again the best example. The lineages of amphipods that colonize the Hadal zone are not distributed randomly across the amphipod family tree. They come predominantly from a subset of the crustacea that already lived in cold, oxygen-rich, high-pressure conditions, the bathyal zone, at depths of 200 to 4,000 meters before making the final descent into the Hadal. The membrane lipid chemistry, the TMAO accumulation, the protein variants, these did not appear suddenly at Hadal depths. They were already present in the bathyal ancestors as adaptations to the conditions of the upper deep sea, and they became the starting material from which the more extreme Hadal adaptations were elaborated. The colonization of the Hadal zone was not a single leap into the impossible. It was the last step in a gradual deepening, each step made possible by the adaptations that the previous step had driven. This pattern of extreme adaptations being elaborated from pre-existing moderate adaptations, rather than arising de novo in the extreme environment, is one of the most consistent findings in the evolutionary biology of extreme environments. It is seen in the evolution of high-altitude organisms, of desert specialists, of organisms that colonize acidic or thermally extreme environments. The organisms that successfully colonize extreme environments are almost always the organisms that were already living at the edge of those environments, that already had the beginnings of the necessary adaptations, and that needed only to extend those adaptations further in the same direction. The Mariana Trench was not colonized by organisms that had never experienced pressure. It was colonized by organisms that had spent millions of years in the deep sea that were already partially adapted to pressure, and that carried in their genomes the raw material for the further adaptation that life in the Hadal zone required. Understanding that evolutionary trajectory from shallow water to bathyal to abyssal to Hadal, each stage building on the adaptations of the stage before reframes the question of how life can exist in the most extreme environment on this planet. The answer is not that life found a way to survive where it should not be able to. The answer is that life arrived at the Hadal zone already holding most of the tools it needed, refined those tools under the specific selection pressure of extreme depth, and produced, over geological time, the community that is there today. The strangeness of the Mariana Trench, the darkness, the cold, the crushing pressure, the organisms that should not be able to exist is real, but it is the strangeness of a place that life has had millions of years to make its own, and the specific sounds that that place produces, and how the organisms that live there perceive and navigate them, is a story that belongs to the next part of this account. Because in the complete absence of light, the physics of the trench do not simply impose silence. They create something that no surface organism was built to navigate. Something that has been shaping the biology of the Hadal zone since before the first vertebrate ever descended into deep water, and that remains one of the least understood features of the most extreme environment on Earth. Sound is the dominant sensory medium of the deep ocean. This is not a poetic claim. It is a physical fact that follows from the same properties of water that make the deep ocean opaque to light. The absorption and scattering of electromagnetic radiation in water limits the useful range of any optical signal to meters, while sound and mechanical pressure waves traveling through the water propagates over distances of hundreds and sometimes thousands of kilometers before dissipating. The deep ocean is, from the perspective of physics, a world built for sound. It is also, from the perspective of biology, a world in which organisms that rely on vision are severely disadvantaged, and organisms that can generate, detect, and interpret acoustic signals have an enormous functional advantage over those that cannot. At the bottom of the Mariana Trench, the acoustic environment is unlike anything in the shallow ocean and unlike anything at the abyssal depths above the trench. The Challenger Deep is not silent. It is bathed in sound, in the continuous background noise generated by the geological processes that created it, by the distant rumble of ship traffic thousands of meters above, by the creak and groan of the tectonic plates whose collision maintains the trench's depth. Below this background sits the biological acoustic environment, the sounds generated by the organisms that live there, going about the business of finding food, avoiding predators, and finding mates in conditions where no visual signal travels more than a few meters, documenting that biological acoustic environment has proved extraordinarily difficult, partly because recording at Hadal depths requires equipment that can withstand the same pressures that challenge every other instrument deployed there, and partly because the background geological and anthropogenic noise is intense enough to mask signals from small organisms. The first comprehensive acoustic recordings from Hadal depths from within the Mariana Trench itself, rather than from instruments lowered from the surface and retrieved before reaching the bottom, were made by a team from NOAA and the Scripps Institution of Oceanography in 2016. They deployed a titanium-encased hydrophone rated to survive the pressure at the bottom of the Challenger Deep and left it in place for approximately 23 days. What the recording captured was not silence. It was a soundscape of remarkable complexity. The low-frequency roar of a typhoon passing over the Pacific surface 11 kilometers above. The sound of a magnitude 5 earthquake originating at a distant location, its seismic waves converted to acoustic energy as they passed through the ocean floor and into the water column. The distinctive signature of a baleen whale, call from an animal thousands of kilometers away whose vocalizations had propagated across open ocean and arrived at the hydrophone as a clear, recognizable signal. And beneath all of this, continuous, the sound of ship traffic, propellers, engines, the industrial noise of the global shipping industry, penetrating 11 kilometers of water to be recorded at the deepest point on Earth. What the NOAA recording did not capture clearly was the biological soundscape of the Hadal zone itself, the sounds made by the amphipods, sea cucumbers, snailfish, and other organisms that actually inhabit the trench bottom. The reasons are partly technical. Small organisms generate low-amplitude sounds that are easily masked by the background noise, and hydrophones, sensitive enough to detect them, require low-noise recording environments that the trench, despite its depth, does not fully provide. But the recordings did reveal something important about the acoustic environment of the Hadal Zone that bears directly on the biology of the organisms living there. The sound velocity in the water at the bottom of the Mariana Trench, determined by the temperature, salinity, and pressure of the water, creates specific acoustic propagation conditions that differ from those in the water above. The trench acts, in certain frequency ranges, as an acoustic waveguide, a channel that traps and propagates sound more efficiently than the surrounding water. Signals generated within the trench are focused and intensified by the geometry of the trench walls and the sound velocity profile of the water. Signals entering the trench from above are attenuated and distorted as they descend through layers of water with different acoustic properties. The biological implication of this acoustic environment is significant. Organisms that generate sounds within the trench are, in effect, broadcasting into an acoustic channel that amplifies and focuses their signals over distances that would be impossible in the open water above. If Hadal organisms communicate acoustically, and there is every reason to expect that at least some of them do, given the absence of any other long-range communication medium, the geometry of the trench itself provides a natural amplification system. The trench walls, the pressure gradients, the temperature structure of the water, all of these physical features shape the propagation of sound in ways that an organism adapted to the Hadal environment over millions of years might, through selection, have learned to exploit. The amphipods of the Hadal Zone present a particularly interesting case. Amphipods in shallow water are known to produce sounds, mechanical clicks, and scrapes generated by the movement of their appendages, used in various contexts, including communication with potential mates and interactions with competitors. Whether Hadal amphipods produce similar sounds, and whether those sounds serve communicative functions in the extreme pressure environment of the trench, is a question that has not been definitively answered. The mechanosensory systems of amphipods, the hair cells and pressure receptors that detect mechanical vibrations in the water, are adapted to function at high pressure in ways that parallel the adaptation of their membrane, chemistry, and enzyme systems. The sensory cells have modified structures that maintain their mechanosensory function under the same pressures that would disrupt the function of surface-adapted sensory cells. Whether those adapted sensory cells are used primarily to detect prey, avoid predators, or communicate with conspecifics, or all three, is unknown. The snailfish that live near the TMAO depth limit, around 7,000 to 8,000 meters, have been observed using the lateral line system, a mechanosensory organ present in all fish that detects pressure changes and water movements in the surrounding environment. In surface fish, the lateral line is used for schooling behavior, predator detection, prey location, and in some species, acoustic communication. The lateral line of deep-sea fish is generally more sensitive than that of shallow water relatives. An adaptation to detecting the faint pressure signals generated by prey movement in a dark environment where visual detection is impossible. In the snailfish filmed at 8,336 meters, the lateral line is visible as a row of pores along the body surface. What those pores are detecting in the acoustic environment of the deep trench what signals are passing through the water and stimulating the sensory cells, is not recorded in the video. Seismic activity adds a layer of complexity to the acoustic environment that has no parallel in shallow marine habitats. The Mariana Trench is geologically active. The subduction of the Pacific Plate generates earthquakes, not only the large surface-felt earthquakes that occasionally make the news, but a continuous background of microseismic events that produce low-frequency vibrations propagating through the seafloor and into the water column above it. These vibrations are part of the ambient sound environment of the Hadal Zone, present at frequencies below the range of human hearing, but within the detection range of organisms equipped with appropriate mechanosensory systems. Whether Hadal organisms have adaptations specifically related to the detection of seismic signals, whether they use the acoustic information in the seismic background to orient themselves, to locate food, or to sense changes in the geological environment, is entirely unknown. The question has not been systematically investigated, partly because investigating it would require long-term acoustic monitoring at Hadal depths combined with detailed knowledge of the sensory biology of Hadal organisms, and neither the monitoring infrastructure nor the biological knowledge currently exists. The chemical senses of Hadal organisms may be at least as important as their mechanosensory systems, and possibly more so. In the dark, high-pressure environment of the trench, where mechanical signals are attenuated by distance and visual signals are absent, chemical signals dissolved. Molecules diffusing through the water provide a communication medium that requires no active signal generation on the part of the receiver. The detection of chemical gradients allows organisms to locate food sources, identify mates, recognize predators, and navigate the spatial structure of the trench without any dependence on light or sound. Chemosensory systems, the olfactory and gustatory organs of marine invertebrates and fish, are generally well developed in deep-sea organisms, as would be expected in an environment where chemical signals are among the few reliable sources of information about the surrounding environment. The hadal amphipods demonstrate this chemical sensitivity dramatically. They are attracted to food sources, the baited traps that researchers use to collect them from distances of many meters, navigating toward the bait by following chemical gradients in the water. In complete darkness, under 1,100 atmospheres of pressure, these small crustaceans detect and follow dissolved organic molecules diffusing from a food source that they cannot see, hear, or touch, arriving at the source within minutes of the trap being deployed. The chemosensory organs responsible for this navigation antennae, bearing specialized chemoreceptor cells, are among the most elaborate structures on the amphipod body, reflecting the evolutionary premium placed on chemical detection in an environment where it is the primary means of locating food. The integration of mechanosensory and chemosensory information in hadal organisms represents one of the more sophisticated aspects of their neurobiology, and one that has received relatively little attention from researchers who have been more focused on the molecular adaptations to pressure than on the functional sensory biology of the organisms. The nervous systems of hadal organisms are adapted to pressure in the same ways as their other proteins through TMAO accumulation, through modified membrane lipid chemistry, through pressure-stable enzyme variants in the neural tissue. But the specific functional consequences of those adaptations for the sensory processing of the nervous system what the hadal amphipod actually perceives when it detects a chemical gradient, what information the snailfish's lateral line is extracting from the pressure fluctuations in the water around it. These are questions that require a different kind of investigation than the biochemical analyses that have dominated hadal research so far. They require observation, direct, sustained, patient observation of living organisms in their natural environment, the kind of observation that has transformed the understanding of shallow marine ecosystems over the past half-century, and that the hadal zone has received only in the most fragmentary and intermittent way. Every camera deployment to the trench floor has produced observations that challenged existing assumptions. Every longer deployment, the baited camera systems that remain in place for hours rather than the minutes that a crude submersible can spare, has revealed behaviors that were not predicted by the biology of shallower organisms. What a sustained, long-term observational presence at the bottom of the Mariana Trench would reveal about the sensory world of hadal organisms, about their behaviors, their interactions, their daily rhythms in a world without day or night, is something that can only be guessed at. The acoustic recordings from the Challenger Deep revealed one more thing that has not been adequately discussed in the literature on hadal biology. The ship noise, the propeller sounds, and engine noise of the global shipping fleet was audible at the bottom of the Mariana Trench, clear and consistent, arriving from every direction, penetrating 11 kilometers of water with enough intensity to be recorded by a hydrophone sitting in the sediment. The deep ocean is not isolated from the noise of the world above it. It is connected to that noise by the same acoustic propagation physics that makes sound the dominant long-range signal in the ocean. And if ship noise can penetrate to the bottom of the Challenger Deep, it can interact with the acoustic environment that hadal organisms depend on. Whether that interaction represents a meaningful disruption, whether the organisms of the hadal zone, adapted over millions of years to a specific acoustic environment, are affected by the novel acoustic energy introduced by modern shipping, is a question that has barely been asked. It is a question that requires knowing first what the natural acoustic environment of the hadal zone is and how organisms use it. Neither of those things is yet adequately known. The sounds of the deep trench, the geological background, the distant whale calls, the industrial noise from above, and beneath all of it, the unrecorded biological soundscape of the hadal community constitute an environment that has been shaping the sensory biology of the organisms that live there for millions of years. That environment is changing in ways that are difficult to measure precisely and that cannot be reversed. And the organisms whose sensory systems were built for a different acoustic world have no evolutionary mechanism for rapid adaptation to the new one. The pace of evolutionary change is measured in generations. The pace of change in the ocean's acoustic environment, driven by the expansion of global shipping and industrial activity, is measured in decades. The gap between those two timescales is where the problem lives. And it is a problem that can only be understood, let alone addressed, by the kind of deep, sustained observation that the hadal zone has not yet received from a scientific community that is only beginning to understand what it is missing. The biology of the hadal zone, taken as a whole, the biochemistry, the ecology, the biogeography, the sensory systems, the evolutionary history, points toward a conclusion that is not the conclusion that the physical conditions of the place seem to invite. The extreme pressure, the cold, the darkness, the isolation these features suggest a marginal environment, one where life clings on with difficulty at the edges of the possible. What the biology reveals, instead, is a place that has been fully occupied by life, adapted to it in extraordinary detail, structured by millions of years of evolutionary pressure into communities of organisms that are not marginal but specialized. Specialized in ways that make them uniquely capable of functioning in the most extreme physical environment on this planet and uniquely incapable of functioning anywhere else. The Hadal Zone is not a frontier at the edge of the habitable. It is its own kind of center, a stable, ancient, biologically rich environment that the rest of the ocean has been surrounding for tens of millions of years without ever fully entering. What produced that specialization, the specific geological and evolutionary history that created the organisms now living at the bottom of the Mariana Trench, is a story that the evidence has assembled in fragments from the few samples and recordings and camera deployments that have reached the trench floor. What remains of that story, in the fragments that have not yet been collected, is far larger than what has already been told. And the direction from which the remaining fragments will arrive, the specific scientific and technological developments that will finally give the Hadal Zone the sustained observation it requires, is not yet clear. What is clear is that every time the access has increased, even slightly, the biology has exceeded what was expected. Every additional camera hour, every additional sediment core, every additional acoustic recording has added something that the existing picture did not contain. The Hadal Zone has not yet exhausted its capacity to surprise, and the next part of this story approaches what lies ahead not from the organisms themselves, but from a direction that the biology of the trench floor has been pointing toward since the first organisms were collected from it. The question of how the trench came to be, what it is, how long it has been that way, and what the answer to that question implies for how long life has been solving the problem of extreme depth. The Mariana Trench is approximately 50 million years old. The subduction zone that created it began forming during the Eocene epoch, when the configuration of the Pacific tectonic plates shifted in ways that initiated the collision between the Pacific and Mariana plates, and began the slow geological process of drawing down the seafloor into a trench. 50 million years is a long time by human standards. It is a moderate time by geological standards. And it is, for the purposes of the biology of the trench, a very long time, long enough for multiple cycles of colonization, extinction, and recolonization, long enough for lineages to diverge from their surface relatives to the point of full genetic isolation, long enough for the molecular adaptations that now define hadal life to be assembled and refined through thousands of generations of selection at extreme depth. But the Mariana Trench is not the oldest trench on Earth, and the Hadal Zone did not come into existence 50 million years ago. Subduction zones have existed throughout the history of plate tectonics, and trenches deep depressions at subduction zones, where ocean crust descends beneath lighter continental or oceanic crust, have been a feature of the ocean floor for hundreds of millions of years. The specific trenches that currently exist are geologically recent. The organisms in them are not. The lineages that colonized the Hadal Zone did so at various times in geological history, some of them long before the Mariana Trench itself formed, in trenches that no longer exist because the subduction zones that created them have since shut down as the tectonic plates continued to move and the geometry of their collisions changed. This means that the evolutionary history of Hadal life is not confined to the history of any single trench. It is a history of repeated colonization of lineages descending from shallower water into successive trenches as the geography of the deep ocean changed over geological time, and of periodic extinction and recolonization as old trenches disappeared and new ones formed. The Hadal fauna of today's trenches carries within it the evolutionary memory of those earlier colonizations. The genetic signatures of ancestors that lived in trenches that are now gone, that adapted to pressures in environments that no longer exist, and whose adaptations were passed down to the lineages that eventually colonized the trenches of the modern ocean. The fossil record of Hadal organisms is almost entirely absent, which is not surprising. The preservation conditions for small, soft-bodied invertebrates at extreme depth are poor. The sediments of modern trench floors are actively subducting into the mantle and will not survive to become part of the geological record. And the trenches that existed in earlier geological periods have been lost to subduction before their sediments could be preserved. What exists instead of a fossil record is a molecular record, the genetic sequences of living Hadal organisms, which carry information about their evolutionary history in the form of molecular clock data. The rate at which mutations accumulate in DNA is relatively constant, which means that the genetic distance between two lineages can be used to estimate the time since they last shared a common ancestor. Applying this molecular clock approach to Hadal organisms allows researchers to ask when the ancestors of today's Hadal species descended to Hadal depths, and the answers, in the cases where the analysis has been done, suggest that the Hadal colonizations are older than the trenches themselves. This is a striking result. If the lineages of Hadal amphipods, for example, diverged from their bathyal relatives before the Mariana Trench formed, then those lineages must have colonized an earlier trench, one that no longer exists. They were Hadal organisms before there was a Mariana Trench to inhabit. When the Mariana Trench formed, they colonized it from the Hadal zone of an adjacent or earlier trench, bringing with them adaptations that had been built in a different geological setting. The Hadal zone, in this view, is not a habitat that life discovered recently. It is a habitat that life has been occupying in one form or another for much longer than any of the specific trenches that define it today have existed. The organisms are older than their current home. This temporal depth of the Hadal community, the fact that the lineages are older than the trenches they inhabit, has implications for understanding the resilience and fragility of Hadal ecosystems. On one hand, the long evolutionary history suggests that Hadal lineages are capable of surviving the loss of a specific trench, that when one trench closes as the plates shift, the lineages in it are not necessarily lost because they can potentially disperse to other trenches or persist in adjacent deep-sea environments until a new hadal habitat opens. On the other hand, the extreme specialization of hadal organisms, the fact that their biochemistry, physiology, and ecology are calibrated to the specific conditions of the hadal zone and cannot function in other environments, means that the hadal zone is not simply another version of the deep sea. It is a distinct environment, and the organisms in it are truly dependent on it. If the conditions that define the hadal zone were to change substantially and rapidly in ways that the geological transitions of the past have not imposed, the lineages that depend on those conditions would have no refuge. The deep-sea mining industry represents one potential source of that kind of rapid change. The Clarion-Clipperton Zone, a region of abyssal plain in the Pacific that is being assessed for extraction of polymetallic nodules, is not itself hadal in depth. But the sediment disturbance generated by mining operations at abyssal depths can produce sediment plumes that travel hundreds of kilometers from the mining site, settling out over areas far larger than the immediate extraction zone. Whether such plumes could affect the food inputs to hadal trenches by altering the chemistry or particle composition of the organic material raining down from above is not known. The models that would predict this outcome do not yet exist, because the baseline ecology of the hadal zone, what it receives from above, in what quantities, of what composition, is not yet adequately characterized. The warming of the deep ocean adds a different kind of uncertainty. The deep ocean is warming slowly, at rates far below the surface warming that dominates the climate conversation, but measurably and consistently. The temperature of the deep Pacific has been increasing at roughly 0.1 degrees Celsius per decade over recent decades. That rate is small, but the organisms of the hadal zone are adapted to temperatures that have been stable for millions of years. Their membrane lipid chemistry and TMAO levels are calibrated to the specific temperature and pressure at their particular depth. They are not calibrated to a moving target. Small temperature changes at depth may alter the pressure-temperature balance that determines membrane fluidity and protein stability, not in ways that are immediately lethal, but in ways that reduce the efficiency of biological functions, reduce reproductive success, and over time alter the composition of hadal communities. Whether this is happening now and at what rate cannot be determined. The baseline data against which change could be measured does not exist because the biological monitoring of hadal communities over time has not been conducted with any consistency. There are no long-term surveys. There is no reference against which change can be detected. There is only the community as it exists now, sampled too rarely, characterized too incompletely, understood too partially to know whether it is the same as it was 50 years ago or different. The contamination of the hadal zone by human-derived chemicals is documented and disturbing, and it arrived before anyone was looking for it. Samples of amphipods from the Mariana Trench collected in recent decades have been found to contain polychlorinated biphenyls, PCBs, at concentrations higher than those found in amphipods from the most polluted coastal seas of China. PCBs were manufactured primarily in the mid-20th century and phased out in many countries beginning in the late 1970s, but they persist in the environment because they are resistant to biological degradation. They accumulated in the tissues of marine organisms, passed up food chains, and eventually reached the deep ocean through the biological pump, the same process that delivers organic carbon from the surface to the trench floor. The amphipods at the bottom of the Mariana Trench are consuming organic material that has been contaminated by industrial chemicals produced on the other side of the planet, decades before the amphipods themselves were born. The Hadal Zone is not remote. It is connected to the surface world in ways that the surface world has not been careful about. The presence of plastic in the Hadal Zone is similarly documented. Pieces of plastic have been found in the digestive tracts of Hadal amphipods collected from multiple trenches. Single-use plastic materials, bags, films, packaging, manufactured relatively recently in the history of the industrial world, have already made their way to the deepest environments on Earth. Incorporated into the marine snow that rains down from the surface and consumed by organisms that evolved in the complete absence of synthetic polymers. How the physical presence of plastic in the digestive system affects the feeding efficiency, reproductive capacity, and survival of Hadal amphipods is not known. Whether the chemical additives in the plastic, the plasticizers, the colorants, the stabilizers that give industrial plastics their specific physical properties are absorbed into the tissues of the organisms that consume them, and whether those absorbed chemicals interfere with the biological processes that Hadal organisms depend on is also not known. The rate at which plastic is entering the Hadal Zone and the biological effects of its presence there are not yet characterized. The data required to characterize them does not yet exist. The trench has been accumulating it for decades. The biology has been absorbing it, and no one has yet measured what that means. What all of this adds up to the PCBs in the amphipod tissue, the plastic in the digestive tract, the warming water, the mining plumes that have not yet reached the trench but that alter the abyssal plane above it, is a portrait of an environment that is simultaneously ancient and fragile, that has persisted through the geological upheavals of 50 million years and more, that has survived the mass extinctions that reset the biology of the surface ocean multiple times, that has maintained a structured and biologically diverse community in the most extreme physical conditions on this planet, and that is now being altered in multiple ways simultaneously by the activities of a species that has barely looked at it. The organisms at the bottom of the Mariana Trench are doing what they have always done: eating, moving, reproducing, sensing the chemical and acoustic signals of their environment, adapted to conditions that have been stable for longer than most lineages have existed. They do not know that the conditions are changing. They have no mechanism to know. They have only the biology they have, assembled over millions of years for an environment that is now, for the first time in their evolutionary history, being changed at a pace that evolution cannot match. The question of what the Mariana Trench will look like in 100 years, or 1,000, whether the amphipod swarms will still be there, whether the xenophyophores will still cover the sediment, whether the community of organisms that science has barely begun to characterize will still exist to be characterized, is not a question that can be answered from the biology alone. It is a question that depends on choices made at the surface by the species that has spent the past century looking up at the stars, while the deepest place on this planet waited, alive and patient, in the dark below. The irony of the Hadal Zone's current situation is that the tools to study it are improving at precisely the moment that the environment is changing. The autonomous underwater vehicles that can now reach Hadal depths and return with video, samples, and acoustic data were not available 20 years ago. The genetic sequencing technologies that allow researchers to characterize microbial communities from sediment samples, identifying hundreds of distinct lineages from a few grams of trench sediment, were not available 10 years ago. The hydrophones capable of recording at Hadal pressure, the pressure-tolerant landers that can be deployed and recovered repeatedly, the computational tools that can process the large datasets these instruments generate, all of them have reached maturity in the same decades that PCBs have been accumulating in amphipod tissue and plastics have been settling into. The sediment, the race between the expanding capacity to observe the Hadal Zone and the expanding set of changes being imposed on it, is not a race that has a clear winner. Both are accelerating. Both are driven by forces that are not easily reversed. What the outcome will be, whether the scientific characterization of the Hadal Zone will outrun the changes to it, or whether the changes will alter the environment before its biology has been adequately understood, is a question that depends on decisions and investments that will be made in the coming decades by people who are, for the most part, not yet thinking carefully about the bottom of the Mariana Trench. The instruments are improving. The access is gradually increasing. The scientific community studying Hadal biology is small but growing. And each expedition to the trench floor returns with something that was not there before. New data, new species, new questions, new reasons to look again more carefully at the deepest place on this planet. The organisms at the bottom of the trench do not share this uncertainty. They are doing what they have always done: eating, moving, sensing, reproducing, adapted to a world that has been stable for millions of years. The uncertainty belongs entirely to the surface. It belongs to the species that looked everywhere except down, that photographed the surface of Mars in higher resolution than the floor of its own ocean, that cataloged the craters of the moon before it cataloged the species of the Challenger Deep. The Hadal Zone has been waiting for that species to pay attention. It is still waiting. And everything that lives there, the amphipods and the snailfish and the xenophyophores and the bacteria in the sediment, is still, as it has always been, indifferent to whether the attention ever arrives. The biology that this story has traced, from the molecular adaptations of hadobacteria to the biogeography of amphipod communities, to the acoustic environment of the Challenger Deep, to the geological history of the trench itself, is a biology assembled from fragments, from the few samples that have been retrieved, the few cameras that have descended, the few recordings that have been made, the few genetic analyses that have been conducted. Each fragment has been scientifically extraordinary. Each has revealed something that was not predicted, and each has made the unmapped portion of the story larger, not smaller, because every new finding expands the range of what might be found in the parts of the trench that have not yet been examined. 50 million years of geological history. The Pacific Plate, still moving, still subducting, still pulling the seafloor down into a trench that life had already learned to call home. Millions of years of evolutionary adaptation. A community of organisms that is more complex, more diverse, and more sophisticated than the extreme conditions of its environment would initially suggest. A sensory world built on chemistry and sound in a place where neither light nor familiarity offers any guidance. A set of molecular solutions to the problem of extreme pressure that evolution has found independently in multiple lineages, in multiple trenches, across multiple geological epochs, and that point, in their elegant convergence towards something fundamental about the range of conditions that life can occupy. And at the bottom of all of it, quite literally, 11 kilometers of water above the sediment of the Challenger Deep, the amphipods, moving through the dark, looking for food. Completely indifferent to the fact that they are, by any reasonable measure, the most pressure-adapted animals on the planet, doing what life does. Because that is what life does, wherever it has the opportunity to do it, even here, at the bottom of everything. If this journey moved something in you, consider subscribing to Sleep on Deep. Every week, we descend a little further. The ocean is waiting.